Thank you very much to the agency for giving this opportunity to me and to the Multiple Sclerosis Outcome Assessment Consortium that I represent here. In this presentation, I will try to give you some, at least partial solutions to all the challenges that Peter talked about when he referred to the impossibility to use placebo in the standard design of clinical trials, at least in relapsing remitting MS. From the study design point of view, as an alternative for minimizing or avoiding the use of placebo, we have basically two solutions. We can run active control trials in which both arms receive anyway an active drug. And I think that next speaker, Luca Massacesi, will address more deeply this topic. Or the second option is to use surrogate endpoints. The use of surrogate endpoints would allow to minimize the number of subjects exposed to placebo and also the time of exposure. So I will focus on the possibility of using surrogate outcomes in MS clinical trials. I will skip all the introduction about MRI and the importance of MRI in MS clinical trials because I think that all of you knows very well the role that MRI had in this last year, at least in phase 2 clinical trials. But this is the sentence that appears in the guideline about the use of MRI endpoints in MS trials. So far, MRI measurements have not been proven to be a reasonably validated surrogate endpoint of the clinical outcomes and are therefore not acceptable as primary endpoints in pivotal studies. I will try to show you a recent evidence supporting the use of MRI as a surrogate endpoint for clinical relapses in MS trials. So this is a pretty old paper that we published on the Annals of Neurology in 2009 that studied the characteristic that a marker should have to be considered a surrogate marker. That is not the correlation with the clinical outcome, but rather the ability to mediate the effect of the treatment on the clinical outcome. In this analysis, we collected all the randomized clinical trials in relapsing remitting MS studied up to 2009, the year of publication, and we put into a correlation the effect detected on MRI lesions with the effect detected on relapses. And as you can see, we found a very high level of correlation between the reduction seen on MRI lesions and the reduction seen on relapses. Following this analysis, many new clinical trials have been run between 2009 and now. So we replicated the analysis in order to try to validate the relationship previously found. And we collected 31 randomized clinical trials in relapsing remitting MS. These were mainly phase 3 trials. This is a weighted regression, what you see. So each ball is a clinical trial, and the size of the ball is proportional to trial size and duration. In this new analysis that has been recently published in the Lancet Neurology, we reevaluated the same relationship. So the black line is the old one, the red line and the red balls are the new one. And as you can see, I was really surprised when we saw the results of this analysis, is that not only the relationship was confirmed, but it was quantitatively confirmed. I mean, the regression line was almost the same. And this analysis is telling us that we can quite precisely predict the effect on relapses in a phase 3 trial from the observed effect on MRI lesions with a simple regression equation. Roughly speaking, for skipping the mathematical details, the effect on relapses is about the square root of the effect on MRI. This is just to have, and this can be really precisely estimated. One limitation of this analysis is that these two effects has been studied concomitantly. I mean, the effect on MRI was evaluated over 2 years, and it was correlated with the effect on relapses over 2 years. What we want from a surrogate is the ability to predict the effect on the longer term. So we selected, performing a sub-analysis, all the couples of trials in which we had the same drug assessed in a phase 2 lasting 6 months with an MRI endpoint, and a phase 3 lasting 2 years on relapse endpoint. And here in this graph, it is reported the effect on relapses really observed in phase 3, the red dots, and the effect on relapses that we can predict from the observed effect on MRI in phase 2. And as you can see, in most of the situations, the effects are exactly the same, very close to one another. And anyway, all are within the 95% confidence prediction intervals. So this analysis is telling us that what we have seen on relapses in phase 3 was already there in the phase 2 study. So this analysis confirms that the paradigm MRI lesion relapses seems to be valid through a broad range of treatments. So this observation supports the use of MRI lesions as a surrogate for relapses. This does not mean that MRI lesions are a valid surrogate for any clinical outcome. And in fact, the relationship MRI lesions disability is less predictable and anyways was not assessed by such an analysis. Let me show you an example for summarizing what I mean when I say that the paradigm lesion relapses seems to hold even in situations where the link with disability is less clear. This is an analysis that we recently presented at the ECTRIMS evaluating the effect of lakinimod. As you probably remember, lakinimod had a small effect on MRI lesions and relapses. It was able to reduce MRI lesions of about 40% and reducing relapses about 20%. But it had a large effect on disability progression. The hazard ratio indicated a reduction in the risk of having a disability progression of about 36%. So in this analysis, there's the effect on disability predicted from the effect on relapses, taking the relationship that has been calculated in a previous work. And as you can see, the effect seen on disability, the effect of lakinimod detected on disability was much larger than what we can expect from the observed effect on relapses. Said that, if we examine the relationship between the effect on MRI and relapses, they are still very consistent. The effect of lakinimod that we expect on relapses is exactly the one that we can predict from the effect on MRI lesions. So, my suggestion for changing the guidelines is a call for more flexibility about the use of MRI as a surrogate. So what I mean with more flexibility, I think that MRI can be used in the design of trials testing the efficacy of drugs on relapses. If our aim is to test the efficacy on relapses, we can use MRI, at least for drugs with a known mechanism of action. And with this sentence, I mean, of course, biosimilars, but I think this is a separate chapter because I think that MRI is already accepted for testing biosimilars. But I'm thinking also to drugs like pegylated interferon, drugs that has a supposed similar mechanisms of action. I think that MRI can be used for testing their efficacy. Another field of application we were talking about before is in the pediatric world. Pediatric is a rare disease. We don't have any approved therapy, and I do still think that MRI can be used to test interferon for children. If we detect an effect on MRI for interferon, we can approve the drug in this situation. Another possible application is for assay sensitivity in equivalence or non-inferiority trials. In the guideline, it is expressly stated that equivalence and non-inferiority trial must have anyway a small placebo group to test for assay sensitivity. And I think that MRI endpoint can be used in a very small and short-term placebo group for giving a message of assay sensitivity. Even if, also because if we have an equivalence trials, probably equivalence is assessed against a drug of known and proven efficacy. So just a small assessment of assay sensitivity can be enough. And the fourth application is in adaptive trials. MRI can be very useful as implemented in adaptive trials. I think that Jeremy Chataway will explore the issue of adaptive trials. I will just show you this example that is taken from oncology. It comes from the Daniel Sargent group of the implementation of an adaptive trial using a surrogate outcome that is not still fully validated. This is the proposal of a design. We can have randomization, an interim analysis in which we assess both efficacy on the surrogate and also the assessment of surrogacy. We test the hypothesis of surrogacy within that trial, and if the hypothesis of surrogacy is confirmed, we can stop the trials and assess efficacy based on the surrogate, and otherwise we can continue the trials and assess efficacy on the clinical endpoint. What I want to point out is that if we design a pivotal trial, a phase 3 trial, we memorize the primary endpoint, we cannot conclude and assess efficacy just based on the p-value and on the significance of the effect on MRI. We'd rather think in terms of precision of the estimate. The effect on MRI should be precise enough to guarantee a predicted effect on relapses which is clinically meaningful. So this is a way of thinking more close to the equivalence trials, not just the p-value on MRI. This is not enough. We must have an effect on MRI precise enough to estimate an effect on relapses we are interested in. So this is my proposal of a bit more flexibility for the specific guideline regarding MRI endpoints. So far, MRI measurements have been proven to be a reasonably validated surrogate endpoint for relapses, while have not been proven to be a surrogate of disability accumulation. Therefore, their use as a primary endpoint in pivotal studies can be considered in very specific situations. And finally, this is a call that other speaker talked about for some flexibility for the introduction in the future of new endpoint, because there's a large effort in the world for trying to validating and introducing the clinical trial design new clinical outcome. And that's all.